Okay, so we'll start the webinar now. You can see my slide, I think. Okay. So hello, everyone, and welcome to today's webinar from Fluigent, entitled Microfluidics Innovations for Diagnostics and Cancer Care. We have an insightful session ahead as we explore how microfluidics is transforming diagnostics and cancer care. I'm Joseph Farah, Microfluidic Application Specialist at Fluigent, and I'm pleased today to welcome Professor Mehmet Toner, a leading expert in microfluidics and biomedical engineering. His pioneering work at the Harvard Biomems Lab has made essential and significant contributions to cancer diagnostics. Before we dive into Dr. Toner groundbreaking research, we'll start with a brief introduction to set the stage for the discussion by revisiting the scientific roots and key moments that led the foundation for this microfluidic field. After Professor Toner talk, we will have a Q&A session. So please do not hesitate to send us your question using the Q&A panel at the bottom of the screen. So let's start in order to understand where microfluidics come from, we start way back in the 15,000s, with no other than Leonardo da Vinci. As you can see, and while the concept of microfluidics didn't exist yet, Da Vinci carefully observed and sketches in his note how fluids moved through natural channels. He was even one of the first to describe and name the turbulent flow in his notebooks. These early observations were the building blocks of fluid dynamics, setting the path for future discoveries. So now if we fast forward to the 1800s, and we find two key figures, Jean Poiseuille and Gottlieb Hagen, who independently studied the fluid movement in narrow tubes. Their work led to what we call now the Hagen-Poiseuille equation, where you can see it, describing the relationship between pressure, flow, resistance, and small channels. It's this principle that we still use to design the microchannels in modern microfluidic systems. Afterwards, moving forward to the 20th century, where another technology laid the groundwork for microfluidics. It is microelectronics. The invention of transistors in the 1940s by the Bell Lab enabled incredible miniaturization, and inspired by the way we could pattern electrical circuit into chips, researchers began wondering, can we do the same with fluids? So based on these questions, it gave rise to the first attempts at miniaturizing fluid systems and sparked the beginning of microfluidics as we know it now. In the 1979, the researchers at Stanford University answered that questions about if we can do it with the fluids by creating the first microfluidic chip for gas chromatography this time. It proves that we could shrink an entire fluidic system, just like circuits, into tiny microfluidic chips. This was a key moment that opened the door to biotech and chemical analysis on the microscale. As the field grew afterwards, researchers succeeding in modeling the fluid flow like electricity. Just as voltage drives the current, pressure can drives the flow. The Hagen-Poiseuille equation became the microfluidic equivalent of the Ohm's law. And by this analogy, it gave the engineers and the researchers a new toolkit, allowing them to borrow established circuit theory to design precise and predictable microfluidic systems. Then a huge leap came in the 1990s with the Professor George Whiteside and the introduction of soft lithography. He pioneered the use of PDMS, a flexible and biocompatible material, to build microfluidic chips quickly and affordably. This breakthrough made prototyping faster and expanded the access to microfluidic tools, especially when we are dealing or we want to work in life sciences. So since then, microfluidics has come a long way, and we moved from simple syringe-driven systems to advanced setup with pressure controllers, flow simulation, wider range of materials, 3D printing, and automated flow software. This evolution has unlocked greater control, flexibility, and applications possibilities across science, medicine, and healthcare. So for instance, as you may know, droplet microfluidics allows high throughput screening and self-sequences. Another example, organ-on-a-chip models that can simulate human tissues and for drug testing. Paper-based devices, which we can see it in our daily life in order to make and enable rapid diagnostics and even in most remote areas. All of this shows how microfluidics is reshaping medicine, biomedical applications, and biology, bringing precision and efficiency to the smallest scales. However, despite all these historical sequences from the 1500s to nowadays, and the progress that it makes in microfluidics, we are still having a lot of challenges in this field. For example, there's still no universal standard for building or integrating devices. The microscale, the flow remains tricky to control. The scalability in terms of translating microfluidics prototype into scalable, manufacturable platforms, which can still remains a challenge. And unlike electronics, microfluidics lacks core components like switches or transistors, which can enable or unlock new applications. So overcoming this limitation is key to unlocking the full potential of microfluidics in diagnostics and beyond. One of the pioneers in addressing these challenges is the Harvard BioMEMS lab, directed by Dr. Toner. His team has led efforts to bring microfluidics into cancer diagnostics, especially in detecting circulating tumor cells from blood samples. They are also exploring microfluidics transistors, which, as we explained before, could do with the hope that these transistors can do for fluidics what electrical transistors did for computing, in addition to other innovative research activities. Founder of the NIH BioMEMS Research Center, professor at the Harvard MIT Division of Health Science and Technology, a member of numerous national and international committees, serving on the editorial boards of many prestigious scientific journals and advisory boards of many biotech companies, and with over 200 publications. We are honored today and delighted to welcome Professor Mehmet Toner to this webinar.
Thank you, Joseph, and let me try to share my slides.
Perfect.
It should be fine, right?
Yeah.
Perfect. Thank you so much. Thank you for the invitation. And it's amazing. There are almost 200 people attending. And for those of you who are in the other side of the world, it's evening. And I hope you're enjoying your day and your evening. It's 20 minutes. It's very short. What I decided to do is actually talk about 4 different projects that are at the very beginning. There are only one paper we publish in each one of these. I just want to touch on these projects and mention the brilliant minds behind these projects. And I won't go into details in 20 minutes. I don't have much time for that, but hopefully we could do that during the chat and questions and answers. Typical disclosure, there are several companies exploring various applications of some of the technologies we develop, but these are all managed according to Harvard MGH rules of conflict of interest. Joseph summarized it very well, that nice serpentine of the growth of the field of microfluidics and MEMS, biomems. And it's really the whole idea there was to achieve exquisite sensitivity at a single particle level to drive a number of applications, mostly chemical and molecular applications. But our interest in around 2000 was slightly different. We wanted to actually look into extremely large volumes that are clinically relevant, what we call bodily fluids. And for example, if you take blood and tube of blood has 50 billion cells. It's a non-Newtonian fluid. 50% of them is particles, cells of different, and these are living things. You can't, bang them around too much. But we asked the question, can we get the throughput to look at millions or hundreds of millions of cells or trillions of cells at a single cell resolution? That was the question we asked. And just to remind people, most of you probably don't know, in fact, many people don't know, there are 37 trillion cells in human body, and about 32 trillion of those, about 90% of them are all blood cells. So it's all blood, except, of course, non-mammalian cells, bacteria. I'm talking about 37 trillion mammalian cells in human body. So the clinical applications are not just blood. There's the leukopak, bone marrow, urine, cerebrospinal fluid, bronchoalveolar lavage, ascites, peritoneal washing. These could be up to 5-liter volumes, and with cells and molecules in them that are extremely useful for diagnostic as well as therapeutic. So that was our main focus, and we built this microelectromechanical system that Joseph mentioned in 2004 as an NIH resource center, which are typically funded for 15 years. And that's where a lot of the genesis of a lot of the work we have done with large volume microfluidics or clinical microfluidics. And just to give a quick evolution of one of the chips, and then I'll dive into 4 applications. For example, with the circulating tumor cell chip, these are extremely rare cells, 1 in a billion in blood. We've developed this technology to very precisely isolate the circulating tumor cells. Sunita did the original work in 2007 with posts that are coated with antibody that binds to tumor cells. This was complicated to manufacture. We went to herringbone afterwards, creating advection convection. Again, the surface is covered with the antibody. And then we went to negative depletion. In other words, not going after the tumor cells, but getting rid of all the blood cells to find the tumor cells. Then you're agnostic to the cancer type, so that works for any cancer type. But the challenge is huge, because even in 2 tubes of blood, you have 50 billion cells, if not more, and you need to find 1 cell out of that and get rid of the 50 billion minus 1 cells without losing that single cell. That's where you need the single cell precision. And then we build with help from Stratec, actually dated. We can't do this plastic manufacturing in our labs. And a monolithic chip that can actually do this in a high throughput. At this point, our flow speed is about 20 million cells per second sorting. We can sort 2 tubes of blood in an hour. And then more recently, we went what I call microfluidics on steroids, and where we can sort cells at 300 million cells per second. And we can process up to 1 liter of blood, and and I will talk about that next. And our processors also that look like a Fluigent device built in the lab to become more and more sophisticated and fully automated now, where we can train a technician within a day to be able to run these devices. So let's not dive into application number 1. And we're really interested in early detection of cancer. It will save millions of lives if we could do that in a reliable, dependable, and definitive manner. Not screening. Screening at some point creates more worries because of the false positives. We want to be able to actually find these cells and make sure they are tumor origin and make sure they are coming from a certain organ. And so Avanish Mishra, who's now a faculty at Harvard Medical School, Mass General Cancer Center, and our center, and a brilliant young mind, has developed this ultra-high throughput magnetic sorting device that we originally published in 2020, and then 2025 we actually published more the clinical results. And the idea here is to be able to get a pint of blood, which will be 500 ml, half a liter, or a leukopack, which could interrogate the whole actually blood volume and concentrate the nucleated cells, and then isolate the cells with the microfluidic, what we call a Venice chip. And then once you have that, we look at molecular confirmation of cancer by looking at two assays: aneuploidy, which is a quintessential marker of copy number variation cancer, and RNA-seq, then we could tell the origin of the cancer. I won't go into details of the chip. It's very similar to the prior chip, with the exception that we bring these magnetic lenses right next to the channels to create an extremely high magnetic field so that we could deflect white blood cells that are labeled with magnetic beads at a very high throughput. So we can now process and sort cells at 300 million cells per second. We can find one cell in 200 billion cells. And here you see some of the early clinical applications with different breast cancer, liver cancer, uveal melanoma. And you can see the number of cells suddenly we find, because now we are looking into a very large volume. We can find thousands of these tumor cells. That's pushing the limits for early detection. You can see circulating tumor cells here. You could stain for specific markers on the surface, proteins that for which we have antibody drug conjugates, so you can target these cells, also therapeutic benefits. And but what I want to show here is that the power of this, and this is Shimbo Huang in Dr. Haber and Dr. Shamala Maheswaran's lab, another brilliant young mind in molecular biology. We developed assays where we could take the enriched circulating tumor cells from high throughput chip and find CNV. If any cell has that in your blood circulation, it's a tumor cell. It can't be anything else. So you have a definitive diagnosis, and then you can sequence those exact same cells. That this is, you look at DNA, you look at RNA here from the single cell, and you can tell which organ it's coming from. So it's a definitive test that could do. We have not, we are still in early days for early detection, but certainly the assay and the chip is working as expected with clinical samples. Another application is clusters. In our work to find the quest to find circulating tumor cells, we have also found that there are some clusters, and not only that, clinically, if any patient has clusters, their prognosis is terrible. And this has been reproduced by many other studies now, and these clusters could be pretty large. And John Ed recently started looking into ways to continuous flow isolate clusters, but he really wanted to push the limits to true apheresis, where you need to process 1 liter whole blood in an hour if you want to do apheresis. And here you can see sequence of images of clusters of 7, 3, or 2 cells. So you would spike different number of clusters and look into efficiency. There are some really interesting microfluidic tricks that some of them I will quickly mention. And Lee Zahn, now who's a professor at Purdue University, joined John's innovation efforts. So the idea here, you take blood from a patient, it goes to module 1, which concentrates the blood, but pushes the white blood cells to the side, so we can give 95% of that blood back to the patient. And module 2 actually isolates the clusters based on inertial focusing physics that we published earlier, where the larger cells are deflected more from the wall than smaller ones. Then we can very efficiently sort them and then give the remaining of the blood also back to the patient. And here you see Lee's web of circuit to do this. He called it PANDA, pre-concentration chip, where, as I said, you can see here, these are white blood cells, streak images, fluorescently labeled white blood cells. At the outlet of the chip, it's whole blood going here. You don't see it, but they're all shifted so that we can skim this portion where you see the flow to the cluster chip portion, and the 95% of the rest is all red blood cells. Give it back to the patient. When you do plasmapheresis, you can't, from a sick person, you can't bleed them to death. And this is the chip that he built. And the beauty of this, there is no change to the blood. That's what you want, to be able to give it back. And he spiked cells in one test case, for example, to 100 ml blood, only 24 cell clusters, only 2 cell clusters occasionally lost, 3 and 4 bigger clusters, almost 100% efficiency to capture them, 90% recovery in 6 minutes. So now his device can actually do 1 liter in an hour with an efficiency of 90% recovery of clusters. Another area that I want to show is, again, another brilliant young scientist, is a professor at Carnegie Mellon, Dennis Heaven, and it's putting things into cells, not only for CAR T cell therapy, the image I have here, but for many different things. People need high throughput and large molecules into the cells. Small molecules, electroporation we can do, but high throughput, billions of cells, and putting things like RNP for chimeric antigen receptor treatment, large protein complexes, it's very difficult to get in. And the genesis of this goes back to a few years, but the idea is high throughput intracellular delivery by viscoelastic mechanoporation. The idea is very simple. What Darren did is inertially focus the cells so they all come at the same location. They all see exactly the same fluid mechanics. It's not like Leonardo da Vinci's crazy things moving around. They're all coming in the same streamline. They are lined up. And then there is a tiny portion, about 100 micron, where we put a constriction. The fluid is viscoelastic, so it stretches that. It doesn't touch the walls. It's not squeezing. It's virtual squeezing. And then it bounces back immediately. It decelerates. And here, actually, with advection convection, the large molecules jump into the cell. And here you see the actual chip. And here you see the cells inertially focused coming to the active region, I call it here. They spend only 1 millisecond there, stretch like this, that porates the cell membrane, it bounces back. And the amazing thing is the particles are moving at 20 meters per second. This is, they're moving fast. And only 1 millisecond in the active region, 4 mil per minute per channel, 200 to 300 million cells per second processing. We can process it at 10 to the 7 cells per mil, almost tissue density. And I won't go into details of the results. He published this, but you need viscoelastic stretching. If you don't put the polymer, you don't stretch the cells as much, you don't get permeation. Small and large molecules equally effectively go in, and membranes close within 5 seconds. So if we put a large dextran, after we porate to see when it stops getting in, after 5 seconds, no dextran goes in. Small dextran takes another 20 seconds. So small pores take a little bit longer to close. But because of this, it's very gentle. It's only 1 millisecond exposure to active region. It's a brilliant way they're designed to porate cells in an extremely high throughput, where we could actually porate the trillions of cells within hours. And the final example in the last 4 or 5 minutes I have is the liquid transistor equivalent. It's yet another brilliant person, Kostav Gopinathan, and he challenged the dogma, and most people thought, yep, microfluidic transistor has been built before. Well, no, not really. And if it was, you couldn't get this published in Nature. And many digital systems are built that could do transistor similar functions. But the definition of transistor is amplification. Nobody has built microfluidic systems that are analog operations. Occasionally, some digital operations like chemosystem and so switch valve transistors are built for digital operations, but not for analog operations. So that's not true transistor. And we are coming back to Leonardo and we're Poisson here again. And if you're in a collapsible tube, there's a number called Shapiro number as a fluid mechanician at MIT. As you increase the flow rate, because the tube is collapsible, it starts collapsing. And at some point, the pressure wave can't keep up with the flow rate, so the message is not sent to the guys in the back. It collapses and reaches a saturation point. That is exactly what a transistor is. And by creating this gate and source and drain and controlling both the gate to source pressure, source to drain pressure, you can create a transistor. And you see these are perfectly electrics analogs of transistor curves, with the exception that it's a pressure flow curve here. And you can see the amplification, you can amplify it. You can see a noisy signal amplify, so you can have a feedback. That is the essence of amplifier. And the gain, intrinsic gain is 22. Now we have solid versions of this. We're pushing up to 100, 120. You can do a regulator. You have an input, noisy input. You can regulate it. Shifter. So you have a, without changing signal, you can shift it. These are all analog equivalents of electric circuits. And you can see the electric circuit and the microfluidic circuit here. But you could also do digital analogs as well. You could do an AND gate where when two things are its maximum, you, in this case, you turn off the pressure. So when these two, bingo, it goes down. When one of them goes down, then it comes back again until both signals are high, then it switches on again. You could do a latch where you could do set and reset, and you can design. So you can actually now, with these 5 important modules, you could build any equivalent of any complex integrated circuit using microfluidics by a priori using the tools of the entire electronics industry. I'm not going to go into too much of the details. We did just one demonstration of loading chemicals into, in this case, cells. We can trap a cell and hold it there. That sends a signal as soon as you block this pressure here. A pressure valve opens and different fluids come here to treat the cells in different ways. And we could, create very complex circuits. This was a simple verification, validation of the transistor in this kind of a cell application. And I think with that, it's 10:30. My time is up and ready for questions. And I mentioned 4 studies, and 6 of the people who were behind those studies, but there are many others who helped to get us to this point. Especially on the biology clinical, Dan Haber and Shamala Maheswaran, Lisha Sequist are wonderful colleagues, friends, and collaborators on the innovation side, which is led by John Ed, but originally Ravi Kapoor was the mastermind of that, to join us from industry to help us build that innovation team. And of course, many resources, funding resources funded the projects, and big thanks to our patients, because we deal with 2,000 to 3,000 patient samples a year. We work pretty much all with patient real bodily fluids, ascites, bronchoalveolar fluids, blood, bone marrow, leukopak, and we developed our tools validating them. And that's the advantage of being a microfluidics lab in a hospital. We try to take full advantage of that. With that, I'm thankful to Fluigent and Joseph for the invitation, and thank you for attending. I'll take the questions.
Thank you, Dr. Toner, for this fantastic talk.
I'll stop sharing. So,
yeah.
Okay.
And all the variety of the applications that you shared. So now, as we can receive your questions, I encourage you to send us the questions in the Q&A panel at the end. And in the meantime, I'll just share in our presentation about Fluigent, just to, in the meantime of sending your questions. So Fluigent, we are an expert on fluid control, especially in the microfluidics and the micro scale. We are a French company based in Paris with the ISO certificates, where whether you are in Asia, US, Europe, or in Africa and all the other, we had rich components of distributors and our presence there. So don't hesitate to contact us if you have any questions in terms of the setup and how you can improve your microfluidic setup. Our main product is the Flow Easy, where you can control the pressure, the flow rate, and high precision compared to the syringe pumps with a high flow rate response and high automation compared to the classical methods. So with this, I will thank you again for attending, and we can start the questions with Dr. Toner. So we will start with the first questions from Kilin Zhang. In one of the applications, you introduced the magnetic fields embedded in microfluidic chip. Can you please explain how you embed the magnetic devices in the chip?
Yeah, it's actually very easy. The magnets are outside the chip, so we clamp the chip into the 4 magnets. But then the channels right next to the separation channel is filled with a magnetic, basically it's a magnetic lens, like a ferrofluid, we fill it in. So the outside magnetic field is concentrated right next to and so that's a very high magnetic concentration next to the channel. That's how we do it, and it's in detail explained in the PNAS paper.
Another question regarding veloporation. Do you see differential uptake based on the mechanical properties of the cells? Could you find a regime where cells would be porated differently depending on the mechanical properties?
Yes, for each cell, we have done, I believe, 7 or 8 different cells, 3 cell lines for primary cells like neutrophils, T cells, and NK cells, stem cells. And each cell requires some optimization to load it efficiently. But the optimization space is fairly well controlled. It's the flow rate, the concentration of the polymer, the type of polymer, polyethylene oxide versus hyaluronic acid, and a few others that we explore. So there's a limited space to optimize, but each cell depends on its mechanical properties, and the size requires slightly different conditions.
Another question where it's a little bit on the trendy now. If in CTC separation, can we use AI or machine learning? Or how do you see it? How we can incorporate this technology to our diagnostics, or for example, specific biomarkers, and distinguish them with high precision?
Yeah, I mean, there are different ways you can, for example, for circulating tumor cells, certainly for if you can get enough of these cells, you can culture and do personalized medicines. In other words, at this stage time of the you need treatment, you can get these cells, culture, and do drug screening. You could monitor the patients. You could look at the response to different things. You could look in the surface proteins. And if you are using an antibody drug conjugate that's going to target certain specific proteins on the cell surface, antigens, you look whether or not they express that before you give the expensive and toxic drugs to people. So that's more for the metastatic. For early detection, we look at this as there are not too many screening techniques with a very high rate of false positives. That means we are scaring millions of people who do not have cancer, but they just don't know. So the idea here, if you are screened with one of these things, such as cell-free DNA, and it's positive, we will see this as a screen. Before you can't do biopsies to everybody, and nor you can put them in expensive imaging modalities and radiate them. And so this will be initially a test, a definitive test, whether or not the person has tumor cells in circulation, early cancer, to see whether or not they need to go for a more invasive test, such as biopsy. In the long run, it might become a screening test if it is robust enough and it is cheap enough.
The question if during the experiments or like the approach that you adopted for with the clusters, if did you see or did you analyze some breaking of the CTC's clusters during the isolation? And how we can
yes and no. It's hard to know for the real clusters because we don't know if we broke them into two small clusters or if there were two small clusters to start with. When we make artificial clusters and spike, we have published several papers where we can tell from the stickiness of the cells. Sometimes if they're very sticky to each other and they don't go through a capillary. Capillary is only 7 micron, much smaller than the cluster. But it's amazing. The cluster unfolds like a string of sausages, goes through the capillary and then refolds back into its cluster. So we have some idea about the physical forces and at what point we can shred them versus they can go through the clog the capillaries or go through the capillaries. The reason why clusters is much more potent, we believe, for metastatic process. They are much less in number, so single cells are still pretty bad because they're more than a few clusters. But the clusters still mimic their in vivo environment, and these are epithelial cells. Because of the cell-cell communication, they don't go through a process called apoptosis. They don't commit suicide. And they circulate in blood, and because they are large, they can clog in a capillary and then grow into the tissue.
A question about what are the biggest challenges in scaling up microfluidic technologies that you experience or you are still experiencing from research prototypes to mass manufacturing?
Yeah, I mean, now, for example, the complex chip I showed you, the Stratec makes for us, it has about 4-meter channels in it, about several million posts that are the size of a human hair. There are about 126 or so parallel units for different functions, so on and so forth. And the reason why it's done in the circular CD shape, because it comes from the Blu-ray technology. They just didn't want to change the shape. Now they have modular other shapes too, but when we did this 7-8 years ago, the cost of that is very reasonable and it's not scaled to millions of chips. Economy of scaling will kick in when these become real products used broadly in clinic. So it will be, my guess at that point, it's going to be $5 to $10. The beads are more expensive than the chips actually. And then the molecular assay is also very expensive. And the total loaded cost of a sequencing plus chip plus beads to separate cells right now to us is, let's say, $1,000 to $1,500, which is even in this current level, it's very reasonable. If you were to do an imaging or biopsy, the cost will be much higher than that. So it's and in the long run, I'm sure it will be a few $100s. And if you can find cancer early in stage 1, today's treatment, you don't need fancy treatments, are provide cure. So you will save millions of lives.
Mm-hmm. I would like your feedback or the regulation or standards that must be met to test microchips with human cells when you designed it or when you start the project with this.
Right. And the only FDA approval that I know of for a microfluidic chip is by the company Dino Di Carlos started in Cytovale with AJ Shaw, who was a graduate student in our lab. And they got the first microfluidic FDA approval for a test to predict sepsis. So there's going to be more of these. Many people are looking into, and so I don't see a huge barrier there because they're quite gentle compared to standard centrifugation or other bead selection techniques that really This is continuous flow, it's scale. And from a manufacturing, I think Stratec and a few others really solved that manufacturing bottleneck. So I don't see huge hurdles any longer for large volume clinical sample processing and applications that need those kind of samples.
Mhm. And we saw like during the progress of microfluidics with the new technologies, one of the technologies, especially like helping prototyping, is the 3D printing. Do you think it is viable or like it would be viable to design, manufacture using these recent, especially if you start with the 3D printing and so on, compared to the methods?
Yeah, I mean, I have a perspective on that. I don't know if it is the right perspective, but I'm happy to share. And 3D printing is going to be always much lower and more expensive than injection molding or any plastic manufacturing technique that's already scaled. So the way I look at 3D printing is if you can do something that is enabled by 3D printing. In other words, you couldn't just do with simple, good old-fashioned manufacturing techniques, further advanced to apply to microfluidics. But if you have to have three-dimensional characteristics that injection molding or other current manufacturing techniques couldn't do, then it becomes, of course, imperative that you use something like 3D printing. But if you think you're going to replace some of the current techniques, I don't think it's going to be feasible. It's going to be always, even the fastest ones, it's going to be expensive. And but I might be wrong, and the time might prove me wrong as 3D printings get better and better. I'm not putting down 3D printing. I'm just saying find applications where 3D printing enables your application, as opposed to trying to mimic what I can do today by other means.
A question about the last application with the transistors. So what do you think, or what are the next major steps in advancing my microfluidic transistors and in order to adopt it with the community? How do you envision their role in the, let's say, next generation biomedical devices?
Yeah, I mean, for the community to adapt it, the best thing that could happen, people will start using publishing papers and advance it. So it can't be just from one lab, Whiteside's it the PDMS, but then we all use that, and that's the power of other people using, discovering new things, discovering difficulties, discovering new applications, advancing new ways of making things. And so Transistor is published recently, and I know there are a few people already in touch with us, and we helped them to replicate the results, and they were able to, and some actually with 3D printing. And so we feel comfortable with the results, and we are hoping that other people use it for their applications. And the big deal for us is to make this in a more, not PDMS, because PDMS is a bit flexible. Transistors require that each work exactly the same way. So we're looking into ways to do this in more solid devices, which we are working on that, on the manufacturing aspects, and building more complex circuits like op-amp and other things. And we are working on those things too. So it's baby steps, but applications we believe is going to be, because the power of it, in my view, I'm not an electrical engineer, but in my view is that all these microfluidic chips has zillions of, like the one I showed you for cluster chip, too many lines. Here you can actually print the computer and the external controls onto the chip. And so you have one inlet, one outlet, a pressure measurement, and you can control the whole thing, and which makes it very powerful. And you can also design using the tools of electronics, these circuits, and then manufacture. So you have a predictable manufacturing or application development platform.
We have a lot of questions, but unfortunately we'll end with the last one for our young attendees. What advice would you give to early career researchers who want to progress in the fields of microfluidics or biomedical engineering?
Jeez. I and one thing it's if for you're asking for academic investigator type people, there is a lot of what I call Me Too type research where you're trying to do what others have done and compete with that. Always looking into new territories and breaking into new grounds. In year 2000s when we are late comer to microfluidics. We weren't there. I wasn't there at the beginning, but in late 1900 and 1995, but really in early 2000s, we got into it. And the vision was very simple. Everyone is going smallest volume and one cell. We said, we're going this way, trillions of cells, real non-Newtonian bodily fluids, but still single cell resolution. So that was the challenge we put in front of ourselves. And so what I'm saying is, don't look for Me Too applications and trying to outdo the next guy, but see if you can walk into new territories. Like PDMS when George Whitesides did this 10 times in many things, including PDMS, but those kind of applications, or David Waite's droplets and so on and so forth.
With this great advice, I would like to thank again Dr. Toner for his talk and for you to joining us today. Thank you for being there and stay tuned for the next webinar from Fluigent. And don't hesitate to contact us if you have any further questions or inquiries. Have a nice day, evening, and see you soon.
Thank you, Joseph. Thank you, everybody.